I'd like to thank the organizers of the conference. Today, I'll be talking about neurons and a little bit about how your brain works. In particular, we'll be using super resolution microscopy, where the ability, as you look in the microscope, is now down to a few nanometers in length scale instead of the diffraction limit of about 250 nanometers. We'll be looking at cultured neurons. Most importantly, we'll be using small quantum dots, where probably many of you are familiar with quantum dots. They're unusually bright and photostable. They're little small semiconductors, but in fact, they're not so small. They're typically about 20 nanometers in diameter, and that has caused tremendous problems with getting them to specifically label. So what we've done is we've developed some smaller quantum dots, about 10 nanometers, and that, I will tell you, gives a very different picture than one gets with small quantum dots. Okay, so what does short-term memory look like? Okay, I know many of you are hungry, and so you want this to be very short. Okay, so I have to do it. A little test first, okay? Close your eyes. Close your eyes. Raise your hand if you think I have a beard. Okay, put your hands down. Now raise your hand. You can put your hands down. There we go. Raise your hand if you think I don't have a beard. Wow. Okay, open your eyes. You did very well. There you go. So I can tell you that for all of you, your hippocampus is working very well. Your hippocampus is a region of your brain which is primarily responsible for short-term memories. And your memory did pretty well. What is a memory? That is the ultimate question. That is, memory is a series of proteins in some neurons. But there are lots of neurons. There's even a lot of neurons in the hippocampus. And there's lots of proteins in there. And a few of them are active. So can we see this little activity which indicates that you've got some memory? But in fact, even in neurons, only part of the neurons change. The part that we're going to emphasize is called synapses, the connections between two of the neurons. And for that, in order to really focus in on synapses, you're going to need the subdiffraction limited resolution and accuracy. Okay, so let me just spend a few minutes more on the biology. What the synapse is, you have nerve 1 connected to nerve 2. In general, in your brain, you have about 100 billion neurons. I mean, think about it. Each of you are pretty smart. You have 100 billion of them. And each one typically connects via these synapses to about 1,000 other neurons. So that means you have like 100 trillion neurons. Isn't that, I mean, really impressive? I'm walking around with 100 trillion neurons. Wow. Okay, so it turns out one neuron does not actually connect physically to another neuron. There's actually a space in there. It's called the synaptic cleft. So one neuron is firing, it activates to glutamate. The glutamate comes to the membrane and then gets released, goes across this synaptic cleft, interacts with what are called AMPA receptors or NMDA receptors, and they undergo a conformational change which causes the second nerve to actually fire. Now this cleft is quite small. It's only at 20 to 40 nanometers in size, and it's filled with proteins and so forth. The synapse over here, right here is the postsynaptic density. Each one has a nerve, which is the presynaptic density, which is coming in, interacting and forming a synapse there. Okay, the size of this is fairly small, about 500 nanometers. So if you want to actually look at an AMPA receptor or an NMDA receptor actually moving around, you're going to need better than this resolution. Furthermore, what we're interested in doing is understanding how a few is becoming a memory, that's you getting memory or going away and you forgetting about it. In order to do that, you need to be able to look for a long period of time. Regular fluorophores don't do that. Regular fluorophores, at least at the single molecule, last only a few seconds, so they're not very good. Quantum dots were recognized as being very good because they can be potentially completely photostable. The problem is they're large. They're on the order of 20 nanometers, which is going to make them difficult to fit inside. So these, I'll show you, are not very good. And if you make a small quantum dot, they will be able to fit inside. So first, you will need 3D, because these are actually at all different orientations. You need small and photostable probe. And lastly, we'd actually like to do this on live cells. And then once we have all of that, we're going to determine the position of the AMPA receptor and the NMDA receptor with respect to this postsynaptic density of these proteins, which are tethering down the AMPA and NMDA. Okay, so very briefly, we're going to do super resolution or super accuracy. We start with Fiona, which stands for fluorescence imaging with 1 nanometer accuracy, and we can get 1 nanometer, 1.5 nanometer accuracy in sub-seconds. And the general idea is a single fluorophore looks like this. It has a width which is equal to the diffraction limit of light, 250 nanometers. But if you have enough photons, the center can be determined much more accurately, in fact, to about a nanometer or so. If you want to not only determine where this is, but actually what the distance between these are, you use a very similar technique, in fact, which was given the Nobel Prize to Eric Betsy and others. Okay, if they're close enough apart, they're unresolved. So the way you do it is you use a photoactivatable fluorophore, where basically it's turned off at first. And then what you do with a low-level light, you activate it. One is there. You take Fiona measurement, you get this to about a nanometer accuracy, then wipes away, then you activate it again, another one comes, and you have a computer reconstruction where basically you have resolution of on the order of a few nanometers. Okay? And so that's what we're using. The trick is to use a small quantum dot. What this is it's a regular quantum dot core, which you buy from a company. And what you do is the companies normally put a very large ligand on it to protect it from the quenching effects of water. This thing is very bright if you put it in organic solvents, but you put it in water, you get nothing. The way you do it is you put a ligand around it to protect it from the effects of water. If you put a very large ligand, which is what they've done, but if you shrink it to make a very small ligand layer, you get our small quantum dots. And our ligand layer over here has a sulfur, which binds very strongly to the core, a hydrophobic layer to repel the water, and then you have some polyethylene glycol to make it soluble in the water. And so the bottom line is it comes on the order of 6-7 nanometers. And in fact, you can make them with slightly different diameters or slightly different compositions, and you can get ones that are emitting at 527, at 560, 615, 650, and so forth. And just like the commercial quantum dots, they're extremely bright. In this case, actually, it turns out they're not quite as bright. They're about a third the brightness of the commercial quantum dots, but still, it's no problem at all seeing single molecules over here, and in fact, you often have a bunch of blinking on and off. Okay, and so then lastly, you then want to make the thing biocompatible. So what you do is at the end of the linker, occasionally you put a carboxylate on the end, and there you attach a streptavidin or an antibody and so forth. Okay? So first, we're going to start with big quantum dots, just to show that it's not very useful. Remember, at first we were put the blue, which is the postsynaptic density, that is, it's essentially where the synapses are. Then in red is the AMPA. And you see here, actually, it doesn't look so good. Whoa. Here we go. Okay, this is actually a single AMPA receptor. First, you can do it in 3D. And then what you actually do is, this is a time projection. So you can see that the AMPA receptor was not at all staying within one synapse. It was tending to run all over. So for example, you see this over here. And in fact, you might say, oh, okay, it's running all over the neuron, not necessarily in the synapse, but maybe it's over here. Well, it turns out looking at 3 dimensions, you actually show that actually even over here, it isn't doing it. So the point is, to just look in 3 dimensions is an important attribute of this work. Okay, and then, so what you find that 85% of the quantum dots are outside of the synapse, and occasionally, 15%, it does go in the synapse. Okay, so big Q dots are largely incapable of getting in the synapse, and largely that's because we can tell when we use small quantum dots. And here, you essentially just repeat the experiment and notice that you see all of the AMPA receptors are basically stationary. Well, actually not quite stationary. This is a synapse, and notice this is in time what the AMPA receptor is doing. It goes into the synapse, but actually it stays bound, but is walking around in what you call nanodomains. And in fact, almost all of it is inside the synapse. And in fact, while it's in a nanodomain, it is definitely in there, and then here, and you rotate a little bit, and it stays in there. Why or what the purpose of the nanodomains are is not really known. Okay, lastly, what we've done is we've labeled not with quantum dots, but with regular fluorophores. And in general, what we find is that, like the small quantum dots, the regular fluorophores localize very well and are not all over the place. So basically, labeling with small organic fluorophores, you get similar labeling to small quantum dots, and it's not the same as big quantum dots. And in fact, here you can see with the small fluorophores, they're primarily located within the synapse, about 0.5 microns. The same thing with small quantum dots, but in fact, the story looks very different when you come with big quantum dots. The reason I want to make a big point of this is because the quantum dots work in the neuron field has really made an enormous impact. And it's believed that the extracellular behavior of these AMPA receptors are very important. And while it may be important, it's certainly gotten a biased view of things. Okay, lastly, well, not lastly, we've not only done this with the AMPA receptor, but we've done with the NMDA receptor, and this too is in nanodomains. It turns out that the AMPA receptors are somewhat more mobile than NMDAs, and the AMPA receptors cover more space and bigger diffusion. Okay, not only can we look in the course of a few seconds, but actually we can look over the course of many minutes. In fact, what we find is that at t equals 0, it's fluctuating reasonably rapidly right here. And then if we look 5 minutes later, it's actually moved. So it's like these horses running around. For example, if they're running around on the membrane, they're really free. Then they come in here. Turns out that they have a diffusion constant such that they're going about 10 times slower. And then the corral, which the horses are in, is generally moving about, again, another 10 times slower. So you can see it over here. Okay, and then if we've talked about how the AMPA receptors and the NMDA receptors are both in these nanodomains. And we don't really know why. The question is, are they in the same nanodomains or are they in different nanodomains? Okay, so that's what we looked at. So we find that synapses, there are some of them that don't have either NMDA or AMPA receptors. They're about 4%. Some have just NMDA, 20%. Some have only AMPA receptor, about 3%. And 73% have both, green and red, corresponding to AMPA and NMDA. And this is what two of them look like. And you can see that the NMDA are generally in their own separate nanodomains, though in fact there are certain regions which overlap. Okay. So we'll skip these, and lastly, go to the people who do the work. First, this was a collaboration with Klaus Schulten. He, in fact, gave us the programmed to do this original three-dimensional imaging. Bill Green is a neuroscientist up at the University of Chicago. And there are my students who've done the work. There are Ann Kai, who's now a postdoc at UCSF, who's done many of the AMPA work. Sang-Hak Lee done much of the NMDA work. Andre de Tomas, who's done many of the fluorophores, organic fluorophores. Duncan Nail started Pinghua Ji, who is the chemist involved. And of course, there's Lucky. Thank you.
Questions?
I don't know if maybe you skipped this towards the end, but do you know if there's a role for the cytoskeleton in organizing these nanodomains in the postsynaptic density?
That is a very good question, and it's really unknown. We've looked at actin. We're starting to look at 797 and also PSD95, which PSD95 probably interacts with both AMPA and NMDA, and 797 probably with just NMDA. There's also another protein called stargazing, which it's unclear during its phosphorylation or not. It probably interacts with one or both of them.
Actually, I have two questions. First one is, do you see the nanodomain with the Cy3b labeling?
Yeah, absolutely.
You see the same, so it's not a steric effect of
No,
the content of the
absolutely.
Okay, great. And did you try to stimulate the receptor and see if it changes the mobility?
Those are experiments which are undergoing now. They're very exciting.
Thanks to Illinois, that we now have longer than usual lunchtime. In fact, and I appreciate that. So we'll reconvene at 2:20.